to begin session 9 in room 2, and I will manage the session. There are 5 presentations, and first of which will be presented by Dr. Gayretli. Dr. Gayretli is my doctoral student. He is going to complete his dissertation and hard working on his thesis. Now he is going to present a study results about stress factors. As you know, high pH levels around Konya basin is a problem that frequently impairs the physiology of many horticultural plants, including grapes, which is the favorite horticultural product in this area. So his study will be about the physiological response of grapevines to artificially performed bicarbonate induced stress condition. Now I think our time is on. Our session is going to begin. So let me introduce Racing directly. Yes, you can begin, please. Now you can go on.
Thanks, Eros, sir. Bicarbonate induced calcium stress impairs the physiology of grafted or non-grafted prima grapevines in nursery. Environmental stress factors as drought, salinity, and temperature extremes reduce the yield and quality of horticultural plants across the world. Under stress conditions, agricultural production should also be enhanced in order to sustain the food necessity of rapidly growing world population. To supplement the food demands of increasing population, protective cultivation has gained particular significance to protect plants and communities against multiple stress factors. Though the majority of crops in protected cultivation are grown in soil, there has been an increasing interest in the use of soilless culture techniques to reduce soil-specific problems. Roughly 30% of total land area of the world is cultivated soils resulting from high calcium carbonate content and alkalinity. Iron chlorides in calcareous soils and water deficit are prevalent environmental stress in the Mediterranean region. Therefore, in this region, they engender critical economic loss in grapevine production. The experiment was carried out in the Research and Implementation Clubhouse of Selçuk University. For the study to hocam bilgisayarımda bir sorun oluyor ama bir saniye.
Bizde problem yok, no problem.
Ekran sürekli değişiyor.
Ha, değişiyor. Ha.
Bir saniye hocam.
Ya buradan devam et, evet.
For the study 2 years, of grapevines were investigated through certain physiological reactions such as leaf temperature and stomatal conductance. Investigations were carried out with daily monitoring after each application tiring 3 consecutive days with a portable parameter. Leaf chlorophyll content was estimated with spread-meter readings of third and fourth nodes of each shoot. For the pH quantification, soil samples in the middle of growth period were collected from the root zone of the pods. Result and discussion. pH and EC values of the experimental growth medium underwent gradual increase during the study. The initial pH value of the medium was 7.6 and displayed significant increase in response to sodium bicarbonate application depending on the solution concentration. The pH values ranged from 7.8 to 8.5 at the end of the study. Similar increase in EC values were also detected as influenced by different dose of sodium bicarbonate solution. Leaf temperature response of grapevines were quite similar after the first and second sodium bicarbonate applications. Therefore, the investigations after the third application were used to compare the effects of sodium bicarbonate applications on temperature of the mature leaves. As can be seen in Fig 2, 100 milliliter sodium bicarbonate for applications result in obvious decrease in leaf temperature. The changes in leaf temperature were statistically insignificant for the vines grafted on 41B rootstock. However, leaf temperature underwent significant decrease after the second treatment in on-root grapevines, depending on the dose. Stomatal gas exchange was investigated with 3 consecutive days following each sodium bicarbonate application. The GAs immediately decreased just after the first sodium bicarbonate application, as depicted in Fig 3. In grafted vines, 50 milliliter sodium bicarbonate applications was ineffective on GAs, while the 100 milliliter dose led to significant decrease for both 3 days. Response of the own-rooted vines were quite different from the grafted vines in that the lower dose was also effective on the GAs after the second and third days of the first sodium bicarbonate application. The GAs values after the second sodium bicarbonate application also displayed significant variations depending on the dose and grafting. In grafted vines, 100 milliliter sodium bicarbonate application significantly decreased the GAs, although 50 milliliter did not result in remarkable change in GAs. In on-rooted vines, dose-dependent decrease was detected following the sodium bicarbonate application. It should be underlined that the GAs values of the vines of 100 milliliter application at third day were not as far below as those of the first and second days. These may indicate adaptive strategies of the grapevines during the prolonged period. After third sodium bicarbonate, dose-dependent decrease in GAs observed in both grafted and non-grafted vines, but the negative effect magnitude of 100 milliliter sodium bicarbonate in grafted vines was not as high as those of non-grafted vines. This may be due to the high tolerance feature of 41B rootstock to high pH conditions. As illustrated in Fig 6, Leaf chlorophyll contents determined after the third application decreased in response to the gradual increase in pH due to sodium bicarbonate. Conclusion. Experimental knowledge on the physiological response of grapevine to calcium stress would aid the growers to recover growth imbalance in nursery. Sodium bicarbonate-induced high pH in growth medium adversely affected the physiology of grafted or non-grafted Prima grapevines in nursery, depending on dose. Decrease in stomatal conductance was more apparent than those of leaf temperature, indicating the sensitivity of gas change of stomatal aperture. In certain cases, physiological constraints were slighter in grafted vines than non-grafted vines. This proves the convenience of the use of 41B rootstock for calcareous conditions. Best culture is with culture. Thanks for your listening.
Yes, I would also like to thank Mr. Gayretli for your presentation, and we are going to perform the second one. The second presentation will be performed by Dr. Masitova. Is Dr. Masitova here?
Hello, yes, I'm here.
Ms.
I'm trying
Ayak?
to share my screen. Yeah. Can you see my presentation?
I couldn't see you, but I could hear. Yes, I am going to introduce you, and I see your presentation. Yes, this presentation. Yes, accelerated method for extraction flavonoids from tarragon, wormwood. Yes, doctor, welcome to our presentation session, and you can go on, please.
Hello, everybody. My name is Aychuruk Majitova, and I'm an assistant professor at the Department of Food Engineering at Kırgız University. The title of my presentation is Accelerated Method for Extracting Flavonoids from Tarragon Wormwood, or Artemisia dracunculus. At first, I want to introduce you the relevance of this research. Considering the fact that plant waste contains valuable extractive substances, chemical process seems to be the most rational direction of its use. Extractive substances are potential sources of flavonoids, natural dyes, anthocyanidins, antioxidants, polyphenols. These compounds are used in the pharmaceutical, food, perfumery, and the cosmetic industries. The most important classes of biologically active substances in the herb and the leaves of tarragon are essential oils, coumarins, flavonoids, phenol carboxylic acids. Tarragon, or Artemisia dracunculus, also known as estragon, is a species of a perennial herb in the sunflower family. It's widespread in the wild across much of Eurasia and North America, and is cultivated for culinary and medicinal purposes. On the right side of screen, you can see the photo of the tarragon, or Artemisia dracunculus. Artemisia dracunculus is a popular seasoning plant used both in fresh state, general leaves, and after it has been dried herb. Tarragon raw material acts as a cholagogue-spasmolytic, antihypoglycemic, and antibacterial agent, and it increases the secretion of digestion juices. It also is an antioxidant and anti-diabetic. The aim of the research work is to optimize the recovery of flavonoids of Artemisia dracunculus by fractional extraction with shorter time periods. Since Artemisia dracunculus is a rich source of flavonoids. As a material for experiments, the following materials were selected from local plant materials: The first one was ripe total mass of containing stems, grains with leaves, and unripe total mass of Artemisia dragonculus and the leaves of tarragon wormwood. So sample preparation was included these steps. First, all samples were air dried to constant weight at room temperature. Afterwards, they were crushed and sieved to observe the effect of grinding. They were sifted in a sieve with different sizes. Afterwards, the samples were extracted, and the extraction was made into different methods: single extraction and 3 times extraction. The total flavonoid content of crude extract was determined by the aluminum chloride colorimetric method using the UV visible spectrophotometer Specort 50 of analytic genome. And to observe the effect of different factors on on flavonoid yield, we made different types of different parameters. We observed. We examined different parameters on extraction, effect of grinding, degree of raw material, and the effect of the ratio of raw material to extractant, and the effect of ethyl alcohol concentration, and the effect of extraction time was examined. Effect of grinding degree of raw materials showed on this diagram. According to this diagram, we can see that the highest extraction yield or the highest extractive substances were observed in leaves. And the effect of sieve size, we can show that in all samples, 1 millimeter sieve size is the optimal sieve size, was selected as the optimal sieve size. On the results of the effect of ratio of raw materials to extractant, we can show that the highest extractive materials were observed in the ratio of 1 to 30 and 1 to 50. In this diagram also, the highest extractive substances was obtained in leaves, afterward unripe total mass, and the lowest extractive materials were observed in ripe total mass. Results on effect of ethyl alcohol concentration. We can show that the highest extractive materials were obtained with 70% of ethyl alcohol, and effect of extraction time with a single. Here the results of single extraction, and the single extraction was made at different times. 15 minutes, 30 minutes, 60 minutes, 90 minutes, 120 and 180 minutes. And the highest extractive materials were obtained as in 120 minutes and 180 minutes. Afterward, after that, we obtained that the highest extraction time is positively affect the extraction yield, and we want to examine the proportional extraction type. Therefore, in this diagram, you can see the single extraction and the 3-time extraction results. As you can see, the highest extractive content, extractive materials or flavonoid content were observed in 3 times extraction with an interval of 30 minutes. It has a positive effect that proportional extraction is positively affect the flavonoid extraction of flavonoids from tarragon leaves, unripe total mass, and the total ripe total masses. According to the obtained results, we can conclude the optimal concentration of extractant is 70% ethyl alcohol, and the optimal ratio for materials to extractants were selected as 1:30 and 1:50%, 1 to ratio. And the optimal degree of grinding of sample materials for all samples, as we see above, 1 millimeter mesh size. And the results of the study have shown that fractional extraction with 30-minute intervals increase the extraction yield of flavonoids. Afterward, we can recommend that extraction with fractional extraction will result in higher content of extractable substances. And thanks for your attention. If you have any questions, you can write to my email.
Yes, Dr. Masitova, I also would like to thank for your nice presentation and also for your on-time completing. Your presentation was about methodology of extraction phenologies, phenolic compounds. Is there any question we can take now and also then after the presentations? I think there is no problem or there is no question for this now, maybe later. The next one will be presented by Dr. Kurt. Yes, Dr. Kurt, you can go on, please.
Thank you.
I think he is uploading his presentation. Yes, his presentation is in silico characterization of sucrose synthase genes in higher plant spaces. Yes, Doctor, please go on.
Okay, thank you. Let me introduce myself first. My name is Fırat Kurt, and I'm an assistant professor in the Department of Plant and Plant Production Technologies in the Faculty of Applied Sciences at Muş Alparslan University. As you indicated, my presentation title is In Silico, or the bioinformatical characterization or identification of sucrose synthase or SAS genes in higher plant species. Let me give you a concise information about the SAS proteins or genes or the enzymes. SAS proteins or the enzymes usually encoded by the SAS genes and the sucrose synthesis have a quite important role in carbohydrate metabolism. The conversion of sucrose into fructose or the uridine D-phosphate into uridine D-phosphate glucose is achieved by the presence of these enzymes or the proteins. They can be detected in various subcellular organs like cellular membrane, cellular intracellular structures, and cytoskeleton. And the functions of these enzymes is not just restricted to conversion of one sugary compound into another one, but also they play quite prominent roles in sugar import, environmental stress responses, nitrogen fixation, and arbuscle maturations. If you're not familiar with the word, the arbuscle, what arbuscle means, arbuscle is like a tuft of hairs or cilia, or a branched of tree-like organs, like found in fungi and etc. And I think this kind of extrusions give some kind of adaptations to the cells within their environments. And the other important role that these proteins play is their contribution to the zinc strength. The sink organs, as you know, like potato tuber or the carrot roots, maize kernels, pea embryos are the sink organs. And these organs, in order to continue on their growth, development, or the maintenance, they have to import metabolites into their environment. And the sauce proteins acting in this pathway as well. The sauce genes in plants have different roles because they are subject of the gene copying process. The sauce genes are copied within the genomes, and after we call it the paralogous relationship. And as a result of this paralogous relationship, they increase they win different kind of functions in order to make plants more adaptable to their environment. If we give some examples for that, for example, Arabidopsis and rice have 6 distinctive active sauce genes, like ATSAS1 and ATSAS2 are regulated in environmental stress responses. ATSAS5 and 6, the orthologs of ATSAS5 and 6 are available in many plant families or the species. In rice, again, SAS1 usually found or detected in elongating tissues, whilst the SAS2 usually found in seedlings germinated and grown under hypoxic conditions. SAS3 and SAS4 was also found in the Caryopsis, and the Citrus anshui has non-allelic SAS proteins like SID-SAS1 and SID-SAS-A, which were differentially expressed in various organs of plant bodies. The objective of this study is to characterize using computational tools, namely in silico analysis, to identify and characterize these genes from different plant species. I used 50 different plant species in order to help for any researchers who may be interested in the gene family, so they can get the information as ready. And as material and methods, as I said, I used the computational tools, and there are, for example, the physiochemical features, properties of the genes were detected: ProtCrem, Phytosome, PIFM, Wolfsort, or Cello servers, or the programs. Some of them are websites. The homology modeling, that means the 3D structures of proteins are achieved, are modeled by using Swiss PDB Weaver, and the quality check of these models were completed by using or the employing the rank page servers. The Motif analysis is completed on the MEME server, and the angle causation sites were completed on the NetNGlic1 server. Let me show you the results. Since I have been employed almost 50 plant species, we have a quite long table. I'm not going to go each one, so just let me, talk about a little bit the columns, what they stand for. Number of amino acids are the length of the proteins. Molecular weights are the weights of the molecules of proteins, and the theoretical isoelectric point is the situation where negatively and positively charged of the negative and positive charges are equal on a protein. So theoretical, in this table, theoretical isoelectric point important for us to understand what kind of pH these proteins act in. And also total number of negatively and positively charged residues, namely asparagine, glutamine, and arginine and lysine, were calculated or predicted, and this gives information about how they are going to how probably they are going to interact with the other proteins inside the cell or the intracellular or intercellular area, and also predicted anglicalization sites were also predicted to understand if they are open to epidemic regulations beside the genomic regulation. Let me give the results. All SAS proteins have acidic character, and total number of negatively charged residues higher than the total number of positively charged residues in all SAS proteins, which shows that these proteins usually act in specific, interact with proteins who have specific motifs or the domains. And the predicted anglicalization sites changed between 2 and 7, showing that the regulations is not just restricted to the genomic regulation, but also epigenetic regulation, also an important factor for the regulation of SAS proteins. Sequence analysis, after sequence analysis, the motif and the domain analysis were conducted. The motifs gives us a hint about how these proteins commonly act in a common pathway, but domain analysis gives us hint about their specific roles in specific metabolic pathways. And as a result of this analysis, the sucrose synthase and glycosyl transferases were detected as two important domains. So these SAS proteins have two domains, probably because of the paralogous relationship and also their domain structure. They are quite selective in their in the pathways they work in, and also SAS enzymes' catalytic activity and its structural flexibility is dependent on this two or double domain architecture. And as a last analysis, the homology modeling was conducted. The sequences was extracted for each species, and Swiss PDB we were used for the model generation. After the models were generated, the quality checks of these models were completed. All models showed higher than 96% of model quality, which seems to be quite good for the protein models. The one thing, and let me show you the 3D structures of these proteins. When I analyzed a random 4 samples from the species that I studied, I detected 4 different structures within the same domain. So that shows that they are quite selective in their roles in biological process. As a conclusion, what we understand is that the sequences that were studied in the study showed similarity and similar physiochemical properties and conserved motif structures along with domain structures. And this information can be used for any lab studies who would like to gain more insight into SAS proteins. If you have any questions, I'm pleased to answer.
Yes, Dr. Kurt, thank you very much for your nice presentation, which was related with multi-gene family on SAS genes and environmental stress factors. So gene family analysis is very important for both understanding the plant's family structure and also the response of plants to environmental stress factors. If any question we can take now or also then. I think there is no question. So thank you again, Mr. Kurt. We can go on with Mr. Kaya. His presentation will be uploaded. Yes, his topic is cryopreservation strategies for aromatic medicinal plant species. Yes, Dr. Kaya, you can go on, please.
Thank you. Hi, I am Ergun Kaya from Muğla Koçman University, Molecular Biology and Genetics Department. At first, I would like to thank all of you for attending this presentation. In this presentation, I will try to talk about cryopreservation strategies for aromatic and medicinal plant species. As you know, medicinal aromatic plant species form the foundation of healthcare practices throughout much of many countries. This is particularly true in the case of traditional medicine practices, including codified systems such as traditional Turkish, Chinese, Unani, and Tibetan medicines, and more localized healthcare traditions. Turkey's wild plant species also form a critical component of modern healthcare practices. Some medicinal species are also in demand for their aromatic properties. The use of the oil of Mentha, Thymus, Eucalyptus, for example, appearing in written texts dating back over the 1,000 years. Wild species from an important component of livelihood strategies in all countries, with wide collection of medicinal and aromatic plants providing a critical source of income in many areas. This is particularly true in areas such as the high alpine regions of the mountains, where agricultural outputs are low and there are few other opportunities for income generations. The medicinal and aromatic plant species of plant biodiversity in both nature and agricultural fields have led the flora to look for alternatives in situ conservation. Therefore, cryopreservation is a useful tool for long-term storage of plant germplasm for next generations, requiring only a minimum space to maintain. With increasing interest in the genetic engineering of plants, the preservation of cell lines such as callus and protoplast, and somatic embryos with incomparable attributes is assuming highly significance. Laterally, cryopreservation was reported to offer real hope for enhancing the preservation of endangered and endemic plant species. Just a minute. Cryopreservation aims to protect and storage of biological materials at ultra-low temperature, usually that of liquid nitrogen, minus 196 centigrade degrees, and is considered as an ideal means for long-term storage of plant germplasm. At this temperature, cell division and metabolic activities remain suspended, and the material can be stored without change for long periods of time. Cryopreservation is the only available method for long-term conservation of vegetatively propagated plant germplasm. In the last 3 decades, a number of different cryopreservation protocols, such as classical slow cooling, vitrification, droplet vitrification, encapsulation, dehydration, and encapsulation vitrification protocols have been developed and utilized for germplasm storage. The The choice of the cryopreservation method to attain the highest survival rates is largely dependent on the plant species and tissue type that is being in cryostorage. Slow cooling or controlled rate cooling techniques involve the simple dehydration of plant material before cryogenic storage in liquid nitrogen. This can be done by slow cooling of the plant tissue to temperature of approximately minus 40 centigrade degrees. Encapsulation dehydration method involves encapsulating shoot tips, and then silica gel or airflow is used to dehydrate the beads until the moisture content drops about 20 to 30 % before they are immersed in liquid nitrogen. Encapsulation technique have many advantages such as easy handling, storage, transferring, germplasm conservation of medicinal and aromatic plant species for cryopreservation, exchange of non-contaminated plant material among laboratories, gene banks, and industries. For preparation of alginate beads, the explants are suspended in calcium-free liquid MS medium with 3 to 4% sodium alginate. The next method, vitrification method. This method involves the treatment of tissues in a mixture of highly concentrated penetrating and non-penetrating cryoprotectants such as PVS2. This solution very popular and very applied at different plant species, and followed by rapid cooling in liquid nitrogen in this technique. The droplet vitrification technique is a modification of the basic vitrification protocol that involves placing the samples within a droplet of 1 to 10 microliter of cryoprotective solution such as PVS2 on a piece of aluminum foil before immersion in liquid nitrogen. You can see the aluminum foil and drops. Droplet vitrification has been widely applied to a number of species, including potato, asparagus, apple, another kind of plant species. An advantage of this technique is the very high cooling and warming rates, thanks to the small volume of cryoprotectant solution used and to the aluminum foil strips, as you show in this picture. These avoids the buffering effect of the cryotube and the relatively large volume of cryoprotectant used in the other vitrification techniques, which slow down the freezing and thawing process. Many plant species have been successfully cryopreserved through the development of various cryopreservation methods. As a standard protocol, vitrification and droplet vitrification are widely applied. Fundamental studies looking at the membrane composition, membrane damage, and repair are likely to help to elucidate why some species are cryosensitive and how cryopreservation procedures can be improved for those species. Our current study on cryopreservation of endemic Thymus silicicus, you can see in this picture 3 different mantis species and 2 different Allium sativum. Another name is garlic species with Asian Agricultural Research Institute, Menemen, Izmir. At first, we tried droplet vitrification technique for both species. We obtained up to 80% regeneration rate, viability and regeneration rate after cryopreservation. Our study is still going on cryopreservation of different medicinal and aromatic plant species. Thank you.
Yes, Dr. Kaya, I also would like to thank you for your nice presentation, which was related with cryopreservation, the last technology, I think, for conserving the plant genetic research. Thank you again. Is there any question? If not, also you can ask later. We are going to introduce the last study that will be presented by Dr. Karabel. Yes, Dr. Karabel, you can upload your presentation. Sorry, Karadal. Yes, Karadal, the last presentation. Yes, the title is Growth Performance of Red Swamp. I couldn't see the following. Yes, fed
Dear
with.
Chairman and dear participants, greetings from Izmir.
Yes, go on.
My name is Onur Karadal. I am assistant professor at Department of Aquaculture, Faculty of Fisheries, İzmir Kreatif Çelebi University. Today, I'm going to talk about the growth performance of red swamp crayfish fed with compensatory feeding regimes. First, crayfish, also called crawfish or crawdads, any of numerous crustaceans with the 10 jointed legs constituting the families Astacidae, Cambaridae, and Parastacidae. Some features of crayfish live in sandy and muddy bottom areas. Feeding type is omnivores. Molting for growth, hiding in the shelters during the light phase. Hunting in the nights, resemble to adults when they hatch, and have regeneration. Crayfish commonly consumed as a food or kept in the aquariums as an ornamental animal by humans. Also, crayfish culture amounts have been dramatically increased all over the world since 2010. Economic crayfish species, the most cultured crayfish species is red swamp crayfish with the ratio of 99% in the world. The red swamp crayfish, Procambers clarki, is a species of camberid freshwater crayfish. It is also known as the Louisiana crayfish, or mudbug. It is native to northern Mexico and southern and southeastern United States. But it has also been introduced elsewhere, both in North America and other continents, where it is often an invasive pest. Main diet of crayfish should be comprised of sinking pellets, but they also enjoy some green vegetables and frozen foods in their diet. They are not picky when it comes to green vegetables and can be offered cabbage leaves, zucchini medallions, and shelled peas. As for frozen foods, they happily accept small portions of frozen fish, daphnia, bloodworms, and brine shrimp, Artemia. As a result of irregular feeding, the growth performance of the organism slows down and some health problems may occur, especially in crustacean species. A behavior called cannibalism, the practice of eating the flesh of one's own species, is observed. A regular feeding regime and a feed with nutritional content are important factors that directly affect the growth, health, and color parameters of crayfish. The feeding regime divided three sections like feeding frequency, feeding rate, and feeding time. The feeding frequency contains portional feeding, cost-effective, best growth rate, and regular feeding. The feeding rate is optimum feeding, the cost-effective, best feed conversion, and feeding time, specific features, and time-effective feeding. The regular implementation of feeding management provides a healthy and balanced nutrition. Compensatory growth is identified by being significantly faster than the growth rate of control animals that have not experienced growth depression held under comparable conditions. Compensatory growth is a phase of accelerated growth when favorable conditions are restored after a period of growth depression. Compensatory growth reduces variance in size by causing growth trajectories to converge and is important to fisheries management, aquaculture, and life history analysis because it can offset the effects of growth arrests. The aim of the study in the situation of lack of food, crayfish does not feel safe themselves. This situation directly causes a stress factor. However, how some species react to this stress factor has not been fully investigated. In this study, the effects of compensatory growth on growth performance of red swamp crayfish belonging to the Cambaridae family Procambors genus were investigated and the findings were discussed comparatively. The experimental conditions, this study was carried out in two recirculating freshwater sump systems with external heaters connected to circulation pump outputs of the systems in Faculty of Fisheries, İzmir Katip Çelebi University. Also, electromagnetic air pumps was connected to each system, and air lines were installed in all aquariums. Detailed information about the systems was presented in other oral presentation in this Congress. In the trial, 8 different compensatory growth groups were formed as twice a day, once a day, every other day, 1 day hungry, 2 days full, 2 days hungry, 2 days full, 3 days hungry, 3 days full, 5 days hungry, 5 days full, and 7 days hungry, 7 days full. Also, the crayfish were fed near satiation with out-aqua bottom feed in the specified periods, presenting the next slide. The water parameters, temperature, dissolved oxygen, pH, and conductivity in the systems were recorded with daily AZ combo water tasting meter. Weight and length measurements were carried out biweekly during 14 weeks experiment. Crayfish weights were recorded in bulk with a presence balance. Total and carapace lengths were measured with a digimatic caliper. All data were subjected to one-way analysis of variance when interaction between the factors is found differences. Two-case multiple range test was used to rank groups with using StatGraphics Centrion statistical software. Also, differences were considered significant at 5%. Here you can see the growth parameters of red swamp crayfish fed with experimental feeding regimes, especially the final mean weight of ATT group, the highest. Also, the final mean total length and specific growth rate was the highest in ATT group. And the mean weights of the 280 and 780 groups started to decrease at the week 4, and the lowest weight was recorded in the 280 group at the week 6, 8, 10, and 12. At the week 14, the lowest mean weight was found in the 780 group. The mean weight of the ATT group started to increase from the week 7, and the highest mean weights between the weeks 8 and 14 occurred in this group. For total and carapace lengths, the lowest value was recorded in 780 group after the 4th week. In the GT group, the highest values of both parameters were found. The lowest survival rates between the weeks 2 and 12 were recorded in the 380 group. The highest survival rates occurred in the GT group from the 4th week. At the end of the 14-week trial, a relation has been recorded among the survival rates of the experimental group, as you can see in this slide. In crustaceans, it has been shown that after a period of feed restriction, they exhibit a pronounced compensatory growth when they return to a sufficient food source. But what is the limit to this compensation? Wu et al., working with juveniles of Phaneropaneus sinensis during 10 days of feed restriction, and then feeding them ad libitum under a 30-day recovery period, reported that 12% of the group was able to regain body weight in comparison with the control group. Also, the studies with red club crayfish, Carax coelicarnatus, have reported that most juveniles can tolerate hunger for a certain period of time, but when they are deprived of nutrition, the growth period is prolonged. Weight loss and a gradual decrease in protein content are observed. In other study with the same species, it was noted that growth after the initiation of feeding was faster in starving crayfish and the fastest in hungry crayfish for the longest time, 30 days. So in conclusion, the growth performance of the ATT group were similar to the GTT group and the GT group. As the main output of the study, it has been shown that the ATT cycle is a feasible feeding regime in terms of reducing feed consumption in crayfish cultivation. The current study represents a different perspective and the importance of feeding regimes and cycles on the growth performance of crayfish species should be subjected in the future studies. Importance of the compensatory growth on the carapace coloration, substrate selection, shelter competition, and reproductive efficiency of various
Dr.
crayfish
Karadal,
species
please sum up.
Should be investigated in the further studies. Thank you for your attention.
Yes, the last sentence. I also would like to thank you for your interesting presentation about animal feeding, interesting animal. Now, for few minutes, we can take any questions about the presentation. Yes, Dr. Kaya, I think you have a question.
No, thank you.
No,
No.
thank you.
Hepinize kongremize katıldığınız için teşekkür ediyoruz hocalarım.
Biz teşekkür ederiz.
Evet, ben de teşekkür ediyorum. Uluslararası kongre olması nedeniyle İngilizce sunmaya çalıştım. Katılımcılara sabırları için, sunumları için teşekkür ediyorum.
Hepinize başarılar hocam. Görüşmek üzere.
Teşekkürler.
Fırat
İyi
hocam,
günler.
bir şey mi diyorsunuz acaba? Sesiniz gelmiyor hocam.
Tam metinleri ne zaman göndermemiz lazım?
Tam metin son gönderim tarihi 30 hocam.
Tamam. Teşekkürler.
Teşekkür ederiz hocam.
Kolay gelsin. İyi günler.
İyi günler.
Activity, patient exchange capacity, and selected nutrients and contaminated trace elements. Characteristics of biochar materials will vary depending on the biochar feedstock and pyrolysis conditions. Biochar is derived from a vast range of carbon-rich biomass, wood residue, crop residue, grass, manure, sludge, municipal organic waste. Biochar is gaining popularity due to its favorable effects on soil, which include increase of cation exchange capacity and nutrient cycling, decreases agriculture greenhouse